Hello and welcome. So today's topic is our microanatomy of kidney. This is the second most complicated thing to understand in first year MBBS, so try to concentrate. Kidney is composed of lots and lots of tortuous uriniferous tubules which are densely packed. Okay. Now this entire component consists of two parts. One is secreting part, another is collecting part. Secreting part consists of nephron and collecting part consists of renal tubules, which are actually proximal convoluted tubule, loop of Henle, distal convoluted tubule, then collecting duct, then duct of Bellini. These are things, okay? So one by one I will talk about these things. First of all, nephron. As you can see in this photo, this is our nephron, which is the functional and the fundamental unit of a kidney. Now, the length ranges from 50 to 55 millimeter, and based on their location, we divide this nephron into two parts. One is cortical part, another is juxtamedullary part. Cortical means this cortical nephron presents in the cortical arch of the entire kidney, and 85% of nephron are basically this cortical type. Now, rest 15% are actually this juxtamedullary nephron. Juxta means just near, and medullary means medulla. So, this nephron basically presents in the cortex, but just near to the medulla. Suppose this is the cortex and this is the medulla. So these nephrons presents in this area just near to the medulla. So we call it juxtamedullary. Now, one thing you have to understand that the loop of Henle beginning from this cortical nephron are small, and the loop of Henle beginning from this juxtamedullary nephrons are longer. So this is how you can identify which are juxtamedullary and which are cortical nephrons. Okay, the number of the nephrons are actually depends individually. Everyone has not same number of nephrons in their kidney, but on an average you have to remember that the total number of nephrons are inside of one kidney is 6 million plus minus 2 million. Okay, another point you have to understand that the entire nephron we divide this into two parts. One is renal corpuscle, another is renal tubules. Now, first of all, renal corpuscle. This renal corpuscle is also known as Malpighian body. Okay, remember this point. This renal corpuscle has again two parts. One is glomerular plexus of capillaries, another is glomerular capsule. Okay. Now, this glomerular capsule is also known as Bowman's capsule. Now, first of all, glomerular capillaries. This is a lobulated tuft of capillary plexus or capillary network made by or formed by afferent and efferent arterioles. Okay. Now, almost 50 numbers of intercommunicating lobules of capillaries are present in these glomerular plexus. Okay. This glomerular plexus contains of modified smooth muscle cells and a special type of mesangial cells. Okay. Before I talk about glomerular capsule, you have to understand one thing. This renal tubule consists of proximal convoluted tubule, loop of Henle, distal convoluted tubule, collecting duct, ducts of Bellini. Now, this proximal convoluted tubule is a kind of tube. Now, if I push this tip of the tubule from upside with forcefully, then it will go depress inside. Now, this phenomena is known as invagination. Okay, try to understand. This is the proximal convoluted tubule, and the beginning point or the tip point of the PCT you are getting glomerular capsule or Bowman's capsule. Now, due to this invagination, the tip portion or the beginning portion of this proximal convoluted tubule becomes dilated and invaginated like a concave shape or cup shape. That's why we call it. It is a capsular form, capsule form. Okay. Due to this, we are getting here outer parietal layer and inner visceral layer. Okay, including lots and lots of special type of cells. Now, in the visceral layer of this Bowman's capsule or glomerular capsule, you're going to find a very special type of cell. These are actually discontinuous large polyhedral cells, or you can say stellate shaped cells. This is known as podocytes. Okay, podocytes because it has big foot plates. And now the renal tubules. Okay, so first component is proximal convoluted tubule, PCT. So beginning point of the PCT actually forms the Bowman's capsule. Okay, Now the inner diameter of this PCT is almost 60 micrometer. Now in the function of this PCT, you're gonna get it reabsorbs the fluid along with glucose, sodium, chlorine, amino acids, then bicarbonate, phosphate back to the blood. Now the second component of renal tubule is loop of Henle. Remember, this loop of Henle is actually component of our PCT or proximal convoluted tubule. Now after going some distance, this PCT goes straight downwards towards the renal medulla or medulla of kidney. Okay. Now what happens? After going straight downwards, it makes U-turn and again go back to the upward or back to the cortex. So we are getting a loop, and in the loop we are calling this loop of Henle, which is consist of basically descending limb and ascending limb. Inner diameter of this descending limb is 15 micrometer and the diameter of this ascending limb is 30 micrometer. Now the third component of renal tubule, which is DCT or distal convoluted tubule, which is basically the continuation part of the ascending limb of loop of Henle. Okay, and now we're going to talk about collecting ducts. So this collecting duct passes through these medullary rays of kidney, as you can see in this photo. When it crosses the renal pyramid, it opens into the renal papilla. Okay. Now when it opens into the renal papilla, the distal part of this collecting tubule is known as Duct of Bellini. Okay. So the Duct of Bellini is actually the continuation part of the collecting duct or the collecting tubule. Okay. And this entire length or the entire collecting duct along with the Duct of Bellini consists of two types of cell. One is principal cell, another is intercalated cell. I cell, P cell. P cell actually works as a water absorption or active water absorption due to the presence of antidiuretic hormone, ADH. What is this hormone? How it works? Again, I will talk about this in details in physiology chapter, not here. And this I cells or intercalated cell secretes acid. That's why our urine is almost little bit acidic. In the entire part of the nephron, you're gonna find lots of special type of cells. Among them, first of all, gonna find macula densa. It acts as a osmoreceptor. It senses the presence of sodium chloride in loop of Henle, and it releases very minor amount or small amount of renin hormone. Next, you're gonna find cells of Pulcisen or Lecis cell. Never say Lacis, it's always Lecis cell. Okay, these are actually polyhedral cluster formation cell present in the vascular pole of the nephron and in between the distal convoluted tubule, DCT. And thirdly, you're going to find juxtaglomerular cells. These cells you're going to find only in afferent arterioles. You're going to never find this in efferent arterioles, only in afferent arterioles. Okay, now these JG cells or juxtaglomerular cells releases large amount of renin according to the body situation. Okay, now this macula densa cell, cells of Pulcisen or Lecis cell, and these JG cells or juxtaglomerular cells, these three type of cells combined together forms juxtaglomerular apparatus or JG apparatus. And lastly, fourth type of cell you're going to find is interstitial cells in renal medulla. This cell secretes erythropoietin according to the body situation. What is erythropoietin? When it releases? What is the function? Again, all these things I will talk about in physiology chapter, not here. So these are the basic anatomy or basic microanatomy of the kidney. I hope you understand a little bit. See you soon. Till then, bye.